Hi there, my name is Hansen. I'm a PhD student supervised by Chris Taggart and co-advised by Mark Baumgartner at the Woods Hole Oceanographic Institution. I'm presenting today on some of our work to characterize right whale habitat in the southern Gulf of St. Lawrence. I'd like to begin by acknowledging my many co-authors who helped make this work possible, and also point out that if you have any questions that come up during the talk or we can't get to them afterwards, please feel free to email me at hansen.johnson@dal.ca. North Atlantic right whales are endangered baleen whales that spend much of the spring, summer, and fall foraging in the Gulf of Maine, Scotian Shelf, and the Gulf of St. Lawrence, as far as we know. They meet their high energetic demands with a pretty unique foraging strategy. They feed on flea-sized zooplankton and as a result have the highest predator-to-prey body mass ratio in the known animal kingdom. They also rely entirely on ocean dynamics to aggregate their prey into energy-rich patches that they can eat. This is a screenshot from Whale Map of right whale visual and acoustic detections in the Northwest Atlantic since 2017. It highlights several of the known feeding areas in the region. Some of these habitats have been studied in detail. Work in Roseway Basin, the Bay of Fundy, and the Great South Channel has revealed that right whales target late-stage Calanus finmarchicus zooplankton. In Cape Cod Bay, right whales feed on a mixed assemblage of Calanus finmarchicus and superabundant small calanoid copepods like Pseudocalanus and Centropoges. There's also anecdotal evidence of right whales in other parts of the world feeding on other taxa like euphausids and decapods. Several habitats, such as those south of Martha's Vineyard or in the Gulf of St. Lawrence, are relatively poorly described. The motivation for characterizing habitat associations is that doing so can improve our knowledge of whale distribution and how whales will respond to changes in their ocean environment, which in turn allows us to develop, hopefully, more effective conservation strategies. Today I'll be focusing on the Gulf of St. Lawrence, which is shown here on this map. In the last 40 years or so, right whales were only seen sporadically in the Gulf of St. Lawrence. That seemed to have changed in about 2015 when whales began appearing in relatively large numbers. This roughly coincides with a decreased occupancy in traditional habitats like the Bay of Fundy, as well as a climate-induced decrease in the availability of Calanus finmarchicus in the Gulf of Maine. Since 2017, the same 40% of the population, or about 140 whales, have been observed in the Gulf of St. Lawrence regularly. They're typically observed between May and October in and around the region known as Shediac Valley, which is indicated in the map by the dashed box. Their shift into this region brought right whales into contact with unmitigated risks from fishing gear entanglement and vessel strike primarily, which led to massive mortality events in 2017 and 2019. And our lab group began conducting field work in the region in 2017 to try to better understand right whale prey and habitat in the area. More specifically, our research was primarily motivated by two questions. One, what are the primary prey of right whales in the Gulf of St. Lawrence? And two, what are the temporal and spatial relationships among right whale presence, prey, and environmental conditions? We joined forces with a team of researchers from the New England Aquarium and elsewhere to conduct vessel-based right whale visual surveys in the region. The goal of these surveys was to find and photograph right whales to understand the distribution and abundance of whales in the area, as well as to get ancillary data on their health, genetics, behavior, etc. We also brought along a variety of different oceanographic sampling equipment to conduct opportunistic sampling of several biological and physical variables in the presence and absence of right whales. On the left, you see, I believe it's Megan Kim deploying a 333 micron mesh ring net to collect depth integrated data on zooplankton abundance and community composition. On the right, you see me working to deploy a profiling cage equipped with a CTD as well as an optical plankton counter, or OPC, to measure the size frequency distribution of particles throughout the water column. If you saw Sarah Fortune's talk earlier this week, this is actually the same cage she used to characterize the prey field of bowhead whales in Cumberland Sound in the Canadian Arctic. So we conducted research cruises in July and August in 2017, 2018, and 2019 in this area. Oceanographic stations were conducted on an opportunistic, non-interference basis with the visual surveys. We were usually able to conduct about 3 stations per day, one in the morning, one at noon, and one at night, or in the evening. A station was typically comprised of an oblique Plankton net tow that was preceded and followed by a CTD/OPC profile. The OPC data was only available in 2018 and 2019. Whale presence at each station was defined as when we observed at least one whale within plus or minus 3 hours and 5 kilometers of the initiation of the station. These maps show the distribution of sampling in each year. The gray and red indicates whale absent and present stations, respectively. The black number in the upper left indicates the total number of stations in that year, with the number of stations with whales present shown in red. We were able to conduct a total of 120 stations, 57 of which had whales present. And then the bottom panel shows a time series of sampling efforts within each year. You'll notice, of course, the sampling design is not systematic, which is again because the oceanographic work was opportunistic on these cruises. For the purposes of this talk, I'm going to be pooling the data from all the years, but in the future I intend to do more detailed analyses of variability on a finer time and space scales. So I'm going to walk you through the results from the net sampling. Again, these samples provide information on the depth integrated abundance of the zooplankton community. The net contents were classified to the lowest possible taxa, and then I reclassified them into 16 groups shown on the x-axis of this plot. The y-axis shows the abundance in individuals per cubic meter. I'll point out that this is a log scale spanning 6 orders of magnitude. The red and gray bars indicate whale present and absent stations. The first point to bring up is that the samples are numerically dominated by small copepods, which are perhaps slightly more abundant in the presence of right whales. This panel B just highlights it's the same information, but it highlights the late-stage Calanus species, including Finmarchicus, Hyperboreus, and Glacialis. Glacialis abundance is quite low, and there's no obvious preference for Finmarchicus, but there is perhaps for Hyperboreus, that middle one. This last panel C compares the total biomass of the net samples and suggests that biomass is slightly higher in the presence of whales. There's still a lot more work to be done here to quantify these preferences more rigorously and work out species-specific biomass and energy contributions, but it's a start. Here are the data from the optical plankton counter, which again provides the depth stratified information on the size-frequency distribution of suspended particles. The first plot here shows an abundance profile. The red and black indicate stations with and without whales present. The solid lines show the median, and the shading shows the interquartile range of the, in this case, abundance in 5-meter depth bins. This profile shows peaks in abundance near the surface and near the bottom at whale present stations in particular. Converting these abundances to biomass or a biomass estimate reveals that the biomass of the surface peak is relatively low compared to that of the bottom peak. And that's all because of particle size. This last panel shows the size-frequency distribution of particles in 20-meter depth strata. It shows that particle size tends to increase with depth, and that whale present stations tend to have larger, more abundant particles in the 80 to 100 meter depth range, which likely correspond to these Calanus species, likely Finmarchicus and Hyperboreus. We can take these same OPC biomass data and combine them with the CTD data to visualize the biomass in temperature and salinity space, which is kind of cool. This top panel shows the full depth profile. The color of the points indicates whale presence again, and the size indicates the OPC-derived biomass in those same 5-meter depth bins. The points are placed at the medium temperature and salinity values, while the vertical and horizontal bars indicate the interquartile ranges of each. The gray contour lines are the isopycnals too. Panel B here just highlights the data from below 40 meters. And it shows that the high biomass is associated with, in this case, likely dense slope water. I still need to do some more work to figure out if the slope water intrusions are a source of this biomass, or if they end up there, if the biomass ends up there via other processes. I also applied a very simple logistic regression approach to begin to quantify some habitat associations. I derived a suite of habitat variables from each station and then used each one as an independent variable in a logistic regression with whale presence as the dependent variable. The habitat variables were physical and biological. The physical variables included water depth, surface and bottom density, and surface mixed layer and bottom mixed layer widths. The biological variables included late-stage Calanus abundance, total net biomass, average OPC biomass, maximum OPC biomass, depth of maximum OPC biomass, and the max OPC biomass at in the bottom 15 meters. So the results indicated significant and positive associations between whale presence and variables correlated with depth, which included bottom density and depth of the max OPC abundance. The results also indicated the height of the bottom mixed layer was positively associated with whale presence. And the only prey variable associated with whale presence was the deep OPC maximum. Interestingly, none of the net variables were significant, which I believe highlights the importance of depth stratified sampling versus depth integrated sampling. So in summary, our results so far indicate that right whales are likely targeting a deep layer of large zooplankton, which is most likely a mixed assemblage of Calanus finmarchicus and Hyperboreus. They also emphasize the importance of depth stratified sampling, which makes sense given the way that right whales are foraging. It also appears as though this deep layer is associated with slope water, and there is, of course, the possibility of multiple different feeding strategies occurring within the same habitat, such as deep foraging on large copepods and supplementary foraging on more abundant smaller zooplankton near the surface. And there's still a lot more work to be done to quantify energetics, time and space variation in these associations, and then to compare these results to regional and systematic sampling efforts that are ongoing. I'd like to end by thanking the many people and agencies that have supported this work. And if I can't answer your question during the conference, please don't hesitate again to shoot me an email, hansen.johnston@dal.ca. Thanks very much for your attention, and have a good day.